Dear children, in this module, let us learn about the tools which is needed for recombinant DNA technology. So tools involved are the restriction enzymes. The restriction enzymes called as a molecular scissors. Second is the cloning vectors, which helps the transferring of a alien piece of DNA into a host, and that's called as a competent host. So three tools which is required is the molecular scissors, the cloning vectors, and a competent host. So what are restriction enzymes or molecular scissors? These are the enzymes which cut DNA at specific sites into smaller fragments. So they are scissors, molecular scissors, which cut the DNA at specific sites. They do not cut it randomly. They cut at specific sites into smaller fragments. And they belong to the class of enzymes called as nucleases. In the year 1963, two enzymes which are responsible for restricting the growth of bacteriophages in E. coli were isolated. And we know what are bacteriophages. What are bacteriophages? They are viruses which infect bacteria. So the enzymes which helps to retard the growth of bacteriophages in enzymes were isolated. Of these two enzymes, one enzyme, how did they act? They acted by adding methyl groups to DNA, and thus the DNA was inactivated. And the other enzyme, they cut the DNA, and those enzymes which cut the DNA were called as restriction endonucleases. So more than 900 restriction enzymes which cut DNA have been isolated from about 230 strains of bacteria. Next, we learn about the nomenclature of restriction enzyme, how the name for a restriction enzyme is given. So let us take an example of a restriction enzyme. It is called as EcoR1. So what does E stand for? It stands for genus, the first letter of the genus. The next two letters, second and third, it stands for the species, the prokaryotic species from which the genus and the species, the prokaryotic genus and the species from which they were isolated. For example, EcoR1 comes from E. coli, Escherichia coli. Then what does it R stands for? The R stands for the strain from which it was isolated. So E for the genus, Co for the species, and R for the strain. And what does this 1 stand for? It is a Roman letter, the order in which the enzymes were isolated from that particular strain of bacteria. So order of isolation. So EcoR1 was isolated from E. coli RY13 strain. Okay, E. coli RY13. So from this E. coli RY13, it was isolated. This was the first enzyme isolated. So hence EcoR1. Restriction enzymes are of two types. They are called as exonucleases and endonucleases. What is the peculiarity of these two enzymes? Exo, they remove nucleotides from the ends of the DNA. Anyway, these exonucleases or endonucleases, they comes under the class of nucleases. That means they act on DNA or RNA, nucleic acids. So exonucleases, they remove nucleotides from the ends of the DNA. And what do these endonucleases do? They cut at specific positions within the DNA. They do not cut at random sites. They cut the DNA at specific positions. And they bind to specific recognition site. So they recognize a particular specific positions, and these positions are called as recognition sites. And these enzymes, they bind to a specific recognition sequence of the DNA and cut the two strands at specific points. We know that DNA is two strands. So both the strands are cut at a particular or a specific point. For example, in this we can see the vector DNA, which is having the sequence GAATTC. Again, on the second strand, which is read in the opposite direction also, it is GAATTC. You can see this. So GAATTC in this direction and GAATTC in the next direction in opposite polarity. And this is a foreign DNA. Foreign DNA means the gene of interest, the gene which is having the DNA which is having the gene of interest. In that also it is having a specific recognition site GAATTC which runs in opposite direction, the same in both the strands. And it is cutting at specific points. The arrow has been given over here. It is cutting between G and A on both these strands. G and A on both these strands. So it attaches, the restriction enzymes attaches to the recognition sites and cuts both the strands at specific points. Now let us learn the mode of action of these molecular scissors. I told they act on specific positions on the DNA. So they first recognize a recognition site on the DNA and attach the recognition site and cut at specific positions. Okay, they cut at specific position. So the restriction enzymes, they cut a strand a little away from the center of the palindromic sites. We know what is a palindrome. A palindrome is a word which is read the same in both the forward and the backward direction. For example, Malayalam. Malayalam is the word is read the same from forward and backward direction. So the recognition site for a molecular scissor is also a palindrome. For example, we see this recognition site GAATTC. On the opposite, on the basis of the principle of complementarity, on the other side, it will be read the same in the opposite direction. In this direction, it is GAATTC. In this direction also, it is GAATTC. What does this GAATTC stands for? It is nothing but the base, the nucleotide bases. Okay, so the restriction enzymes, they cut a strand, they cut the DNA strand a little away from the center of the palindromic sites, that is the recognition sequences, but between the same two bases on the opposite strands, that is between the G and A on this strand and between the G and A on the other anti-parallel strand. And as a result of cutting the DNA at specific sites between the same two letters or between the same two bases, what happens? This leaves single-stranded overhanging stretches at the ends. Single-stranded overhanging stretches. These are the single-stranded overhanging stretches. The bond between these two strands, that is the hydrogen bonds, are broken off. And as a result, single-stranded overhanging stretches are present at the ends. And these single-stranded overhanging stretches are called as sticky ends. So these restriction enzymes, they get attached at a specific site, which is called as a recognition site. And this is nothing but a palindromic sequence. A palindromic sequence in a DNA is a specific sequence of base pairs which reads the same in both the strands, in both the strands in the forward and backward direction, when the frame of reading of both the strands is kept the same. That is, one strand runs from the 5 to 3 and the other strand runs from the 3 to 5 direction. So sticky ends, what do? They form hydrogen bonds with the complementary cut parts. This stickiness is facilitated by the action of the enzyme DNA ligase. We had studied DNA ligase where in replication, in which the Ogasaki fragments are stuck together with the help of a molecular glue called as the DNA ligase. So these strands, overhanging single-stranded stretches, it is the same in the case of the vector DNA and the foreign DNA, in which they have to combine together to form a recombinant DNA. So with this help of single-stranded overhanging stretches, they form hydrogen bonds. They form hydrogen bonds when they stick together. Okay, and they are stuck together with the help of the enzyme called as the DNA ligase. When cut by the same restriction enzyme, the DNA fragments has the same kind of sticky ends. Okay, because each restriction enzyme has a particular, or they recognize a particular sequence. So EcoR1 will recognize only the sequence GAATTC and cut between G and A. And when the same enzyme is used to cut the vector DNA and the foreign DNA, the similar stretches are created and they will be stuck together with the help of hydrogen bonds and with the help of the enzyme DNA ligase, and they are joined together by DNA ligase. In this we can see the foreign DNA. This is the foreign DNA and this is the vector DNA. And the same restriction enzyme is used for cutting both the DNAs. And we can see this is the gene of interest, and this here also they are cut at the same recognition site. And as a result, it will generate single-stranded overhanging stretches. And ligation is done. So we can see this is a recombinant DNA. That is a plasmid with a gene of interest is called as a recombinant DNA, and then it is introduced into a host. Imagine this host is a bacterial cell, that is a E. coli. So when this recombinant DNA is introduced into a host, it gets transformed, its phenotype gets transformed, and thus it is called as a transformant. It is called as a transformant. That is, a host has got transformed due to the due to receiving the recombinant DNA. Dear children, in this module we learnt about the first tool, that is restriction enzymes or molecular scissors, and the mode of action of how they are used to cut the DNA strands. Thank you.